right. Well, good morning, everyone. I think we should probably get started at the top of the hour. First off, welcome back, everyone, after a brief hiatus from Nephrology Grand Rounds. It's nice to see all of your wonderful smiling faces. This beautiful Friday morning we have. We have two excellent speakers today, including one of our visiting professors joining us from California. So without further ado, we'll go ahead and get started with our first presenter, Nicole Vo.
Thank you for your introduction. My name is Nicole. I'm acting assistant professor in our division, and my mentor is Dr. Professor Benjamin Friedman. Today, I would like to talk about my project that I'm working for 5 years. It's about developing advanced gene editing methods in human organoids. So everybody here know who is she? She's Jennifer Doudna, is a Nobel Prize winner, and she famous for CRISPR. She brought the tool for all of the scientists and allow us to edit DNA in living organisms. And how about gene editing in the context of our division, nephrology? So I have a chance to write a review paper about CRISPR-Cas9 in Science, and this is the list of the disease in kidneys so far. I have found that scientists use CRISPR-Cas9 to try to learn about the disease and to try to find the treatment for the disease. So gene editing has a huge potential to cure many different kidney disorders. My next question is, how about human organoids? Can it help the genome editing succeed? And the answer is yes. So human organoids, we can use human organoids as a platform to investigate any gene therapy treatment. We can use it as a collaboration for other experimental groups, such as in animal and patients, to investigate dose-dependent quantitative assays. Using this platform, we can measure the editing rate and to measure the toxicity if it occurs in the treatment. As you know, that animal group is very expensive to perform experiment, and also some of the human gene does not exist in the animal. So human organoids, is a unique opportunity for us to perform gene editing. So I started with Vino in Vino lab as a senior postdoc, and I was impressed by his beautiful organoid system, especially kidney organoids. So Vino gave me a very cool project that you need to develop a method to edit kidney organoids. And then you measure the side effect of the edited organoid if it occurs. So how to do this? First of all, I have to set up fluorescent arc system that I can visualize the editing events in the organoid. So basically, I have a cell of stem cells that express GFP. And I introduce CRISPR RNB into the cells. And if the editing is successful, the cell will turn to non-GFP cells. So using this fluorescent arc system, I make kidney organoids from GFP stem cells. And what we're looking for here is the GFP loss region in the organoid to represent for the knockout area. For example, in this LTL positive signal area, we have a huge GFP loss area here. It means that the editing is successful in proximal tubule of kidney organoids. How about monocytes? So yes, we can detect knockout cell in monocyte as well. And you can see that here where we have the monocyte positive signal, but we don't have the GFP signal in the cells. And those monocytes, Dino and I have a joke about it looks like a berries, so we call it edited berries. So my next question is whether this editing event can be detected by other methods like sequencing. In fact, yes. So we have two different kind of organoids derived from two different IBS cell line. And we perform the method into these mature organoids, and we send the sample for next generation sequencing. And the results show that we can detect the editing event at around 15% or 10% in both different type of organoids as well. So the method works in our case. That's it. Like, Bino just give me the project, I make it work. However, when we went to NIH and we meet the director of the program, she gave me a question, okay, whether your method apply for all the human organoids. So to answer her question, I have to start a new project, which is very difficult. So I have to establish different type of human organoids in Vinolab, and I successful. I made a liver, culture liver organoids. I make brain and eye organoids. I make cardiomyocyte or heart organoids. And those organoids I qualify by using specific marker to standard, or I do the quality control by using qPCR assay. So I have the platform, I have the system. I start to think about a new system, the fluorescent on system in those organoids. The reason I think about this because the fluorescent off, in the fluorescent off system, we cannot distinguish the cell death and the cell get edited because when it dead, there's no GFP. So for the fluorescent on, when the cell get edited, it will turn the color for us to see. So we have a cells we call like ionized cells that have no color. And inside the cell, we have a stop cassette, lock, stop, lock. The stop cassette control the expression of the TD tomato gene. And when we introduce CRISPR-RNA into the cell will turn red when it gets edited. So this is the design of the RNA in the ionized cell line at the stop cassette. So you see guy 1, guy 2, and guy 3, and this cell have like SP40PA terminators. So this terminator inhibit the expression of TD tomato. So I designed a guy to attack the terminators and remove it, so the TD tomato gene can express in the cell. So I made it. I edited all of the organoids I made, and you can see here, like in proximal tissue, all the red cells. In the heart organoid, a lot on it, especially in the brain and in the eyes. And the thing made me really, really surprised is how I can make the brain organoids in the kidney lab. Because I think that's maybe, I'm lucky. And you can see that the brain here in the neuroepithelium, we have a lot of red cell represent for edited cell in retinal and RBE culture as well. So then my question is whether this gene editing event can be measured and can be quantified, and the answer is yes. So in kidney, we get around, 8%. In lung, we get around 7%. In heart, we get around 12%. And in liver, we get the highest rate is around 17%. So after I edited all of the organoids, my next question is whether the method, the therapy I developed is safe for the organoids. Because we don't want, like after editing, all of them die. So we want it alive and it functions normally. So I perform several assays like genotoxicity assay, physiology assay, and immunological response assay. In those of the assay, my R&B treatment show no effect in the genome. The organoid after being edited can function well can uptake the dextran, the fluorescent Alexa conjugated dextran, and compared to the MOC or compared to the latroculin is the positive control that inhibits the uptake of the dextran. And my R&B treated organoids also does not promote any immune response. As you can see in the qPCR assay, that if we treated the organoid with interferon gamma, is known for promote the expression of APO1 or ISG15 gene. And the results shown here, both treatments, we don't see any increase of this gene in those organoids. So it shows that my method is safe and I can proceed to the next step. And as you can see here, this is the cardiomyocyte, the heart organoids, and you can see here after the R&B treatment, we can see the heart beating, and especially you can see the red cell beating. And I measure the beats per minute in these organoids, and there's no difference between the MOC treatment and the R&B treatment. So, okay, I answered the question from the NIH director of the program. Yes, we can edit all of the human organoids. And they asked me the next question, whether we can target the editor to the specific cell type. And this topic is very difficult because, so far nobody can do that. And why they ask this question? Because targeting editing can improve the editing efficiency. It can help us to deliver the editor to hard to reach cell types. We can use lower doses and we can spare non-targeted cell from the side effect. So I have a plan with Bino. So we make a plan. Step one, how to do this? Step one, we use kidney organoids and we, as a platform, to screen the molecular targeting agent we call MTA that bind to kidney epithelium. Step number two, we coupling or we tethering the MTA to the Cas9 and optimize the ratio between MTA and Cas9 and B in kidney organoids. And step three, we assess editing efficacy in kidney organoids modeling, especially in polycystic kidney disease. So far, we are successful to edit, to target things on the monocyte. As you can see here, the top panel, where the red cell TD tomatoes show up, exactly like the area of the WT1 positive standing. And this is the mud cells. The results show that we can get around more than 10% in this method. And modo target is possible to target the monocyte in the kidney organoids. How about tubules? Yes, we can do this. As you can see in the top panel, when we use this MTA with the RNB, we can detect the knockout cell at the LTL positive area representing for the proximal tubule. And I measure the knockout area, and I can get around 5% in this experiment. So in conclusion, that organoid can be used to test editors in different stages. We can measure efficacy, safety, and phenotype in human context. Even though the editing rate is quite low in these organoids, and my next step I will do is I want to improve my method to get higher editing rate. And interestingly, the targeting specific cell type is possible in human organoids. So my future work is I want to improve the efficiency of my method in intact organoids, and I would like to collaborate with other scientists to evaluate my tools. That I would like to thank you so much for my PI, my mentor, Professor Friedman, who teach me and give me a very cool project, which I really love. I also thank to Ross Wilson and Doug, who helped me in this project from UC Berkeley. Thanks to Raymond to check my writing, and Scott Michael in Penn State University for another very cool, interesting project about sonication in organoids. And I thank to my lab member and all of the alumni who contribute to this project. And that I'm happy to take any questions. So the molecular target agent is basically the specific antibody that can bind to the receptor, the surface of the cell in the specific cell type. Yes. And in our team, we also have a team to design specific peptides that can help to bring that complex inside the cell, enter the nucleus through the endocytosis process. So thank you for your question. So let's go back to the experiment I did. So this is the MTA with the RNB. As you can see, we can target the monocyte very clearly. It's sharp. It aligned at the WT1 positive signal. However, when we only use the peptide or the RNB, you see the cell, the knockout cell somewhere else, but not in the monocyte area. Or we use the IgG control. It's another mock control, for the antibody treatment. And we see it's also randomly by somewhere inside the organoid compared to the untreated. So I think the limitation where I can improve is probably on the peptide, that we need to design the peptide and the size of the Cas9 probably should be a little bit smaller. That the cell easier to uptake to it. The peptide promotes a little bit more of the endocytosis. And from our part, me and Dino already developed the method to support this process going through the organoid. Yes. So I think in order to treat the disease, we need to get at least 50% of the editing rate. And that's the question for the next hour that me and Dino should write and apply for. Your expectation at the beginning, you didn't mention with me that I have to get 50%. I should make it work. Did try in early stage of organoid development. But we don't like that way because we want to edit directly into mature organoids, where it's already formed all of the structure and the cell type. In that way, we can use the organoid to the next then back into the organ. The early stage can give us more accessible ways to edit the cell. However, it will affect the function and the shape and physiology of the organoid. So that is the disadvantage of the early stage. For the late stage, when the organoid already mature, the organoid already have function. This is the yes. So as you know, kidney is very thick organ. So the, the, the editing rate in kidney is around 10 to 15%. The highest the rate in different organoid type is in the liver. The I think 17 to 20%. Thank you so much.